Assalamualaikum. Hi, and good day to all. So basically, this is our second lecture, and here we will explore some of the advancement made in the area of drug delivery system that utilizing virus as the carrier or as the vector. So virus delivery vectors are one among the many nanomaterials that are being developed as drug delivery materials. So in today's lecture, we will focus on the use of viral vector for gene therapy, vaccine development, and also for cancer immunotherapy. The commonly used virus as vectors, such as adenovirus, retrovirus, will be discussed in detail on their structural properties and how they can be utilized as a carrier. So nanomedicine often involve the use of nanocarriers as a delivery system for drugs and genes for maximizing the therapeutic effect and also to minimize the side effect. From drug administration to therapeutic activity, nanocarrier must evade the host immune system specifically and efficiently target and enter the cell and release their payload into the cell cytoplasm by endosomal escape. This process actually constitute the early infection state of a virus. So therefore, virus are a powerful sorry. So therefore, virus are a powerful natural nanomaterial for the efficient delivery of genetic material. So here, let's have an overview on how virus infect the host cells. So basically, after the entry into host body, virus can evade the host defense and reach specific organs or cells. Once viruses are taken up by cells, they traffic the intracellular space and release their genomic materials to replicate themselves. And then the cell explodes, releasing thousands of brand new virus particles and infect another cell, and so on, and cause the disease. So imagine that what we can do if we can transfer the therapeutic gene by using virus as a vector. So this is a general structure of a virus, and virus size-wise exhibit nano dimension size. It can be ranged from 10 to even to 200 or 500 nanometer, but most of them have like a 50 nanometer in size. So structure wise, the virus, this is the general structure of a virus. So it can contain the envelope, which act as a protective outer layer of the membrane, and they also have the protein coat, or we call it capsid, that protecting the genetic material of the virus. And the genetic material of the virus is actually is inside the core, which encompassing the RNA or DNA, which allow the virus to copy itself and cause infection. And then one of the important structure component of a virus is this capsomere or spike protein, which play an important role on how the virus infect their hosts. This is actually a starting point of virus mechanism in infecting the host cells. For example, like the COVID-19 virus, it has the S spike protein, which initiate the entry into the cells by targeting the ACE2 receptor, right? So this is the function of spike protein. which to initiate the attachment to specific cell surface. So the viral vectors are usually derived from the wild-type virus whose viral genes have been replaced with the heterologous genes intended for cell manipulation. This is the therapeutic gene. So the virus mediated gene delivery systems utilize the ability of virus to inject their DNA inside host cells. And those take the advantage of the virus ability to replicate their own genetic materials. Plus, as we can see in the virus structure, they have the capsid protein, which act as a protection to the genetic material. So in here, if we put the gene of interest inside the virus, it will also be protected by this capsid protein of a virus, just protecting it from like enzymatic degradation and chemical instability and so on. So here is the general characteristics of an ideal viral vector, which means that the virus must be able to enter the desired human cell with high specificity. If required, receptors of the virus must be present on the cell surface, and importantly, it should be able to evade the immune system response so that it will not be destroyed by the immune systems. So that's the beauty of virus. Virus has some way, unique mechanism to avoid being recognized by the immune systems, which can cause infections. So this is what we want to utilize to make it into a gene delivery system. Toxicity is really important. We need to ensure that when we replace the viral genome with the gene of interest, that the genome is completely removed or attenuated, and the genome should not be able to revert back to its virulent state, which means that it is replication defective form. It should be in replication defective form, which means that they cannot replicate. So this will prevent an undesired expansion of the virus within the target organism. And in terms of insertional capacity and production efficiency, every virus have different capacity for DNA insertion. For example, the genomic material inside of the genomic material can just accommodate to certain length of DNA. So we need to choose wisely on the type of virus and how big the DNA material that we wish to insert. And in terms of long-term expression, in here, what we in this wording here is that a viral vector needs to either integrate into a chromosome, which means that for long-term expression, the virus need to have the ability to be able to integrate into the nucleus of host cells for a long-term expression. But sometimes, depending on the type of disease that we want to have the therapeutic effects, sometimes it's not necessary for us to have a long-term expression of the virus. Some disease may require a short-term. So depending on the type of gene that we want to target and how it can be used will be depending on how long we want the gene to stay inside the body to produce its therapeutic effect.